Dr. Matthew Albrecht, when you are ready, take it away.
Okay, great. Thanks, Elizabeth. Sorry for the delay there. So can you see my screen? Am I in full screen mode right now?
Yep. We can see
Okay, perfect. Okay, well, welcome everyone. Thanks a lot for joining the presentation today. And I want to thank the Plant Conservation Alliance planning team for the invitation to speak about our ongoing research with rare plant translocations in the United States. So my talk today is really divided into two sections. The first is providing a background on the practice and patterns of plant translocation in the United States. And then the second part of my talk, I'm going to talk about some recent research we've been doing, sort of looking at the drivers of plant translocation success in rare plant reintroduction projects in the United States. So let's just start off. What is conservation translocation? And I'm using the IUCN definition for translocation, which broadly means the intentional movement and release of a living organism where the primary objective is a conservation benefit. So in the case of rare plants, this is usually being done to improve the conservation status of that rare species. And when we look at broadly translocations, that can also include things like rewilding, which is basically restoring an organism, typically it's an animal, to restore some natural ecosystem function or process. But for today's talk, I'm really focusing on plants. I'm going to really focus on one aspect of translocations, and that's population restoration. So this is any conservation translocation within a taxon's indigenous range. And so that kind of restoration could be to sites where the species exists, and in which case that might be an augmentation or what some people call an enforcement. It could also be to an unoccupied site within the species' historical indigenous range, in which case some people might call that a conservation introduction. But my talk today is going to focus within the historical indigenous range of a species. I'm not going to be focusing on assisted migration or assisted relocation or the other terms to describe translocations outside of a species' geographic range. So if we look at these kinds of population restoration activities, there's really a spectrum of different activities that can occur with a rare species within its historical indigenous range. One of those could be, again, an augmentation. Some example of that is work being led by the Missouri Department of Conservation with Mead's milkweed, Asclepias meadii, where the goal is to try to bolster the size of existing populations to increase the number of flowering individuals in that population. We also may be doing augmentations to increase genetic diversity or genetic variation within populations if there's been a genetic bottleneck. Another example might be to reestablish metapopulation processes. So in that case, we'd be reintroducing to unoccupied sites to try to restore gene flow for species that tend to form metapopulations. Maybe one example could be something like pitcher's thistle, Circine pitcheri. Another example of this kind of reintroduction activity would be situations where you need to rescue and relocate a rare species because a population of that rare species is in imminent danger of destruction. And a great example of that is some work done at Fairchild Botanic Garden with Amorpha herbaceae. So how do these kinds of translocations, and I'm going to use the word translocation and reintroduction and population restoration sort of synonymously throughout this talk. So how does this kind of activity fit into the conservation philosophies for species recovery? And the philosophy I'm going to draw from is really taken from the Center for Plant Conservation's best practice guidelines. And when you think about reintroductions or translocation activities, it's really never the first action to take for a critically endangered species, even when crisis is typically imminent. So our number one priority in species recovery is to conserve species in their native habitats, in situ, in as many locations as possible. And so the first step for species that are in serious trouble, that may be declining precipitously in the wild, really should be threat control, identifying those threats first, and then trying to control those threats, managing the habitat to try to reverse the decline of that species, and then turning to ex situ conservation as well to sort of back up or safeguard those existing populations that potentially are declining in the wild. So these are sort of our first steps that we need to take before we would consider doing a reintroduction or sort of translocation activity with a rare species. So when are conservation translocations justified in species recovery? And again, I'm drawing directly from the CPC best practice guidelines, and I'd highly encourage you, if you're involved in reintroduction activities, to turn to these freely available guidelines at saveplants.org. We have an entire chapter that is basically outlines the steps for you to go through if you're interested in conducting a reintroduction activity. So reintroductions are justified if the species is extinct in the wild, or if several other criteria are met. So for example, in cases where the species distribution is known and there are a few small declining populations, if management activities have happened at those sites and they've been unsuccessful at reversing the decline of that species, or in the case of conservation, you're unable to actually protect the sites that rare species exists in. Maybe they're on private sites and you're just unable to protect and manage them adequately. The other key thing is that we need to know what the threats are to those species, and we need to sort of know how to control them. Otherwise, our reintroductions will likely not be successful if we don't understand the threats that are sort of driving the decline and rarity of that particular species. So the flip side of this is, when are conservation translocations not justified? There are a number of different reasons here. I'm not going to go into all of them, but I wanted you to make you aware of the sort of philosophical guidelines and the CPC best practices and the ones that we're sort of adhering to in our work on synthesizing reintroduction projects across the US. There are a number of different reasons why a reintroduction may not be justified, and most of it has to do with if these criteria are not met, they would lead to likely low success rates with that introduction. So, for example, if you don't have high quality diverse source material and you don't know how to propagate that material, that's going to be a serious challenge in your reintroduction program. If you're unable to deal with existing threats, maybe it's a disease or some other threat, you need to know how to control and minimize that particular threat. Importantly, we don't want our reintroduction activities to undermine the protection of existing sites, right? So we don't want people to think that simply by reintroducing the species to a protected natural area gives us free rein to go out and basically destroy, damage, or degrade existing natural populations if we can just simply reintroduce them. We don't want that to happen, and so we would never want reintroductions to undermine the protection of existing sites. We also don't want to be doing reintroductions when they're not supported, either legally, administratively, or socially. And we wouldn't want our reintroductions to sort of interfere or harm or conflict with other management activities at a site that involve other threatened or endangered species. So this is some work we did a few years ago looking at recovery plans on the Endangered Species Act, and we looked at how often some form of reintroduction activity was proposed as a method for recovery for a species listed as threatened or endangered on the Endangered Species Act. And I broke this up into the sort of conterminous US and then Hawaii. And when we looked at the number of plans that were available, that's the orange bars in this graph, and the numbers are a little bit outdated because this was done about 10 years ago. But I think the overall patterns sort of hold today, even if we were to update this looking at newer recovery plans that have been developed. If we look at the taxa with recovery plans, and then we look at the ones that require reintroductions, just over about 70% of recovery plans for species in the conterminous US had reintroduction as a conservation activity as part of their recovery. In Hawaii, it's over 95% of plans call for some form of, again, augmentation or reintroduction activity to ensure the persistence of that species in the wild. So reintroductions and the sort of spectrum of reintroductions are really an important activity for the recovery of species, and they're increasingly being used to help recover species in the wild. And there are really a couple of classic examples of species that have been recovered and delisted on the Endangered Species Act, in part due to reintroductions. So one example is robin cinquefour, which was delisted in 2002. This is a species known from about 4 locations, and it was recovered in part because, well, hiking trails that went through the natural populations were sort of rerouted, and that mitigated a major threat to the species, which was human trampling. But there were also a couple of new populations that were established that really helped to increase the distribution of the species and provided some redundancy on the landscape in case of catastrophic loss of those existing natural populations. And because of these activities in situ, this species was essentially delisted from the Endangered Species Act. Another great example would be the Tennessee coneflower, Echinacea tennesseeensis, one of my favorite wildflower species. This is endemic to limestone glades in central Tennessee. This was a species delisted around 2012, 2013, and it's known from 5 naturally occurring populations. And within those 5 populations, there are about 36 kind of distinct colonies, which is basically a unique site with plants. And about half of those colonies are introduced colonies. They were introduced from seed from those 5 existing natural population sites. And so this again helped to create redundancy on the landscape. It helped to increase the distribution and abundance of this species, and in turn, along with other in situ measures such as habitat protection, habitat management with prescribed burning, helped to really recover this species, and it since has been delisted on the Endangered Species Act. So if we scale up and look globally, we see that conservation translocations are an increasingly common practice. So if you look in the literature, some handful of papers that have come out in recent years sort of documenting and exploring reintroduction activities across the world. A paper came out a few years ago looking at over 1,000 reintroduction projects in Australia. There's hundreds of documented reintroductions in China. And recently, a team of collaborators developed a database in Italy documenting hundreds of plant translocations there for conservation purposes. So this is really an increasing sort of global activity, and there are thousands of these projects on the ground as we speak. And so this really sort of begs the question of how successful are these translocations and how are they really contributing to rare species recovery? And there have been a couple of papers that have tried to sort of tackle this question. The first is a seminal review published back in 2012 by Godefroy et al. In Biological Conservation, where they looked at close to 250 reintroductions globally. And their general conclusion was that at that time, based on the data they had in hand, they felt like reintroduction was generally an unsuccessful conservation strategy as it was currently being conducted. And that was based on the sort of low survival and reproductive rates of reintroduced individuals in their data set. And their data set was based on about 3 years of data, and I'm going to come back to that point a little bit later. A follow-up review by Sarah Dalrymple et al. Looked at over 300 reintroductions, mostly in Europe and the US, and basically concluded that we don't really have enough information for many of these reintroductions because they're not monitored well enough to be able to answer this question. So there was too much uncertainty in the outcomes of these projects, so they didn't really draw any substantive conclusions about reintroduction as a conservation practice. And finally, in a more recent review in Australia, again, they looked at over 1,000 reintroductions. They found that success rates were highly variable, and they found that it was really species identity was explaining more of the variation in their data set than other factors such as like habitat or ecosystem type or life form and some other factors that they looked at as well. So they saw a lot of variation among species and success rates. So I think this really leads to some key questions in this sort of growing field of plant translocation science or plant reintroduction biology, whatever you want to call it, that need to be answered. And so one of those is, under what contexts are translocated populations most likely to become self-sustaining in the wild? Are there particular life history traits or species characteristics that might influence translocation success? I guess another way to put this is, are some species just more easily reintroduced than others? And finally, sort of what is the importance or relative importance of these sort of species traits the techniques we're using to reintroduce species, and the attributes of the locations and sites that we're reintroducing to. What is the sort of relative importance of these different factors in influencing our reintroduction outcomes? And so this is sort of the question that my research team has been trying to tackle in the past several years by synthesizing a variety of different information. And most of that information is now housed in a database that's maintained by the Center for Plant Conservation. You can find this. This is accessible on saveplants.org. And this data set went live, our database went live a couple of years ago. And it currently has about 480 projects from 187 taxa throughout the United States. And a lot of the data sets we've been sort of putting together over the past decade have ended up in this database. And so we've been sort of drawing on information now in this database to sort of tackle these questions. So I just want to give you a brief overview of this database. This is a great resource. If you're a conservation practitioner and you're interested in doing reintroductions, you can go to the reintroduction registry on the saveplants.org website. You can see what species are being actively reintroduced. And you can create an account and even look at particular information about those reintroduction projects to understand how other people are designing their reintroductions, how they're implementing them. And then, of course, there's contact information there too that you could potentially reach out to people to understand how they're conducting their project, and you can learn from their sort of techniques and the way they've been doing their projects. So it's a really great resource if you're in this field, and I'd highly recommend checking it out. So this is sort of an overview of the projects in the CPC database, and you can see that the projects are sort of heavily concentrated in a few geographic areas in this data set. Not surprisingly, those are the three represented global biodiversity hotspots in the United States, right? So about 40% of the projects are from Hawaii. We have good representation from California in this data set, and then also from Florida, which is in the world's newest declared biodiversity hotspot, the North American Coastal Plain. And you can read more about the CPC database in a paper that's coming out in Applications in Plant Sciences that was led by a postdoc at CPC, Joe Bellis. And this is the distribution of life forms in this database. So most of the reintroduction projects are with perennial herbs, at least in the continental United States. But when we move to tropical regions like Hawaii, that's where we see a majority of the woody plants, woody shrubs and trees are a much greater representation in Hawaii. And if you're sort of new to this field and you're interested in making a contribution, it's hard to find reintroduction projects for graminoids, non-vascular plants, ferns. These are all sort of poorly represented in the CPC database, likely because there just aren't that many reintroductions with these kinds of life forms out there. So for the rest of my talk, I'm going to talk about some results that we have from some work we've been doing on a subset of projects that we pulled out of this sort of larger database that sort of met criteria for us in our analysis. And I should add that almost all these projects are typically well-curated, well-monitored reintroduction projects. So they have really good data associated with them that allow us to sort of draw out variables from these projects and begin to answer some of those questions that I posed in that previous slide. So for the rest of the talk, I'm going to focus on a data set that's about 275 reintroduction projects. It's mostly slight majority of those are from continental US, but we have a significant number from Hawaii. They represent 127 taxa. 83% of those taxa are G1s or G2s. So again, we're looking at truly globally rare species that are being reintroduced for conservation purposes. And all of the events that we're analyzing, all these projects are solely for conservation purposes. The average age of projects in our data set is about 8 years, which is significantly greater than most of the other reintroduction projects that were analyzed previously in other global reviews. So one of the seminal reviews looked at 3 years of data. We're looking at much longer term data at 8 years, and we have projects up to 46 years of data in this database. So they've been monitored periodically for 46 years since they were reintroduced. So we have some really long-term projects that are represented in this data set. For most of the analyses I'm going to present today, we sort of removed annuals and long-lived species from the data set simply because statistically they were problematic in the data set. And I'll sort of mention those later in the talk, but those are just a handful of reintroduction projects. We also removed reinforcements and augmentations for some of the analyses I'm looking at because we couldn't differentiate individuals that were either reintroduced into existing populations from naturally occurring plants in those populations. So we removed those in the cases where we couldn't determine the differences between those. I just want to acknowledge some key collaborators and partners for this project. So 2 postdocs have been instrumental in bringing this data analysis and data sets to fruition. First was a postdoc on my research team, Oyomura Osuzawa Peters, who really helped spearhead assembling this data set several years ago. And then I also want to acknowledge Joe Bellis, who has been instrumental in sort of analyzing this data set and taking the lead on writing a couple of our papers that have come out of this work. I also want to acknowledge my collaborators at the Center for Plant Conservation, Joyce Mashinsky, the former president of CPC, and the current interim president, Katie Heineman, as well. And a number of project partners who submitted data to this effort on their reintroduction projects that I'm including in the analysis here. We thank them for their contributions to this data set. Also, we have a couple papers out that feature data from this that I'm going to present in this rest of this talk. Both of these papers appear in Conservation Biology. The top paper, the bottom paper just came out a couple months ago. Again, this was led by CPC postdoc Joe Bellis, and a majority of the work I'm going to present is really coming from this bottom paper, which is open access and available to anyone on the World Wide Web. So from this larger data set that we put together, we sort of pulled out about 29 different predictor variables from this data set. And we grouped those predictor variables, and these are variables that might be influencing reintroduction outcomes, into 3 broad categories. So the first is species traits or species characteristics. So these are things like, what is the life form? Is it herbaceous? Is it woody? What is its mating system? Is it, obligate outcrosser? Has a mixed mating system? What's the pollination mode? How rare was it? Was it G1, G2, G3, etc.? So these are all sort of species traits or species characteristics. And then we have another set of predictor variables that are basically techniques, and these are variables that are under the control of the conservation practitioner or those doing the reintroduction. So these are things about what kind of propagule did you use? Did you use seeds or did you use seedlings? How many individuals did you outplant? How many years did you outplant individuals at a given site? How many source populations did you use? Did you use mixed source populations? Did you use multiple? Did you use single? What was the genetic relatedness? Was it a single source? Was it mixed sources? What kinds of management did you do at the site? How many different management interventions did you do before and after you planted? So these are all things essentially under the control of those doing the reintroduction projects. And then finally, we have variables that are really related to the location or sites in which those reintroductions are occurring. So these are things about habitat suitability, and we had practitioners rank the suitability of habitat that they were reintroducing a focal species into. What is the ecosystem type? The number of threats at that site? How did threats change over time from the time of outplanting to the time of last monitoring? And then how old is the project, and what kinds of obstacles did the conservation practitioners face in their reintroduction projects? So we put all of these variables together, 29 of them, and we use machine learning techniques to ask which one of these variables were best at sort of explaining different metrics of reintroduction success. And for this talk, I'm going to focus on 4 metrics of reintroduction success. The first is population persistence. So in other words, did that reintroduced population persist over time? The second metric is next generation recruitment. So this is basically, are there new seedlings, second second generation seedlings or next generation seedlings emerging in that new population? Has that been documented yet? And then the third metric is really population size. So how big is the population size at the last monitoring date? If we're working with rare plants, we typically want to have as large populations as possible. So we're looking at what factors influence population size. And then finally, we looked at specifically reproductive population size. So how many individuals were actually reproductively mature at the last monitoring period? You can think of this maybe as a slightly rough measure of effective population size. So again, we put all these variables in a hopper, and for each one of those different metrics of success, we simply ask what were the sort of most important predictors in explaining each one of those metrics. I'm going to show you a series of graphs like this one in the upcoming slides. And so let me just kind of walk you through how we interpret these. These are the top 10 most important variables that will describe each one of these metrics of success in our reintroductions. And again, we group these variables in the three different categories. Species traits or characteristics are yellow. Those techniques under the control of the practitioner are orange. And the variables associated with sites or locations are red. So when we look at what factors explain whether or not that reintroduced population persisted over time, there's sort of three variables that really kind of come out as being important. The first is what we call founder size. This is how many individuals were planted at that site over time. So founder size was the most important. And then that was followed by two metrics of habitat quality. So one is called habitat quality change. So how did the habitat quality for that focal species change from the time of outplanting to the last monitoring period? And again, that last monitoring period is going to differ for each one of these projects. It could be a few years, it could be up to over 40 years in some cases. But we want to know, is habitat quality improving, or is it staying the same, or is it decreasing over time? And that metric turns out to be important. And the other factor that's important is just simply what was the habitat quality at the time of outplanting. And again, these are categorical. This is a categorical variable where the habitat quality could be either highly suitable, had fair suitability, or poor suitability. So those all turn out to be important. When we look at founder size, we see that as you increase founder size, the number of individuals outplanted, this reduces the probability of extinction of that reintroduced populations over time. And in this graph, I'm showing you that relationship, and you see this inflection point kind of at like 100 individuals, just over 100 individuals, where those reintroductions with less than 100 individuals tend to have about a 20% greater risk of extinction over time than those that had more than 100 individuals outplanted. So founder size is really important. And this is something that comes up in some of the previous reviews of plant reintroductions. Founder size is consistently an important variable that comes out of many of these analyses. So again, habitat quality and habitat suitability also turn out to be important for the persistence of these reintroduced populations. So on that first graph, looking at habitat quality change, those projects that had a negative change in habitat quality tended to have about a 15% greater risk of extinction than those that had no change or had a positive change in habitat quality over time. So no surprise there. So when habitat quality degrades, we have a higher risk of extinction in our reintroductions. And the same goes for habitat quality at the time of outplanting. So those projects in which the practitioner said, well, the quality of the habitat that we reintroduced the species into was either fair or poor, those tended to have greater risk of extinction over time than those that were reintroduced into good or sort of excellent quality habitat. No surprise there, but in a large data set like this, it's interesting to be able to pull out from all the noise these trends in habitat suitability and founder size. So the next variable we looked at is next generation recruitment. So again, is there evidence that new seedlings are recruiting into the population? Is there evidence that the founder population is able to successfully sexually produce seeds that then go on and produce seedlings in the population? That's one of the key metrics for identifying a successful reintroduction. And again, we find founder size is the most important variable coming out as important, sort of explaining that metric of success. And again, we see those projects that had more than 100 individuals tended to have greater probabilities of next generation recruitment over time in those reintroductions. So some of our earlier work, drawing on a subset of projects in this data set, looked at what we call the lag time in recruitment. So this is the time period between which the individuals become reproductively mature in your founding population to the time when you first see seedlings emerging in that reintroduction. And for some species, like Lantana canescens, it can happen really quickly. So you might get individuals maturing within the first or second year. They go on to produce seed, and you see new seedlings within a year after that. So you have some species that have really short recruitment time lags, and they tend to recruit very quickly. But we see many species that have these long lag times before they recruit seedlings. So for example, in Pedicactus knowltonii, it was over a decade in some cases from the time we saw individuals flowering in the population to the time that seedlings were first documented to occur. And so these lag times are really important in evaluating success of these projects because you may be monitoring them for many years and you may think, well, this population's not doing well because I'm not seeing any new seedling recruitment happening. It may just take time for many of these species, particularly the longer-lived perennial herbs that seem to have slow life histories and invest little in seed production. They're going to have these longer recruitment time lags. So that's a really interesting trend that we saw in some previous well-monitored reintroductions. And so again, there are certain sort of taxonomic groups that seem to show consistently low and slow recruitment. Mead's milkweed is one example, and then some of the cactus reintroductions in our data set also sort of show this trend as well. So things from like grasslands and more arid habitats tend to show these longer sort of lag times. So our machine learning analysis of this large data set of predictor variables allowed us to look at interactions between these different variables. And one of the cool interactions that sort of came out of this data set was that we found that those reintroductions with larger founder sizes, they tended to have increased population resilience over time as the habitats deteriorated. So let me walk you through this graph on the right-hand part of the slide. So those reintroduction projects that are extinct are in the dark gray. Those that are extant, are in the light gray. And when there's no habitat quality change or the habitat quality change is positive, again, we see a much lower extinction risk or rate in those reintroductions. When we look at those projects that have had a negative habitat quality change, those with larger founder sizes are more resilient to change, and we see a greater proportion of those surviving on the landscape than we do than reintroductions with usually about less than 100 individuals. So larger founder sizes are increasing resilience, and that was kind of a cool interaction that we were able to sort of detect in this data set. So this is looking at another one of our metrics of success. This is population size. And again, we see founder size coming out as being one of the more important, as the most important predictor of total population size at the last monitoring. But something we see different is we're starting to see variable shaded yellow. So species traits begin to be more important in explaining metrics of population size and reproductive population size, as you'll see in the next slide. Whereas in the previous metrics we looked at, next generation recruitment and population persistence, it was pretty much all being driven by techniques and by site attributes or location attributes. So on the right-hand part of the slide, we're looking at the probability of achieving either a small, medium, or large population size, depending on what the initial number of individuals were at the time of outplanting. And so when you have more individuals, you tend to decrease the probability of those populations becoming small over time, and you increase the population of having populations with at least 50 individuals in them over time. So again, larger founder sizes sort of guards against the effects of small populations, those demographic and genetic bottlenecks that can happen in small populations. If we plant larger numbers of individuals out over time in these reintroduction projects, we tend to sort of guard against some of these issues of developing small population sizes. So we looked at reproductive population size and we sort of divided it up into short-term and long-term. So those projects, we looked at like less than 6 years, and then we looked at projects that had more than 6 years of data. And something we saw was that the factors affecting reproductive population size shifted with time. And so when you look at sort of short-term projects, we see life form is one of the most important predictors of how many reproductively mature individuals are in the population. Founder size is not far behind again. But when we look at longer-term projects and we look at reproductive population sizes, we see the number of threats and seed production rate turn out to be sort of important drivers of the number of reproductively mature individuals in those populations. And so what we found in this data set was that the reproductive population sizes varied by growth forms. And we tended to find that temperate perennial herbs with weak seed production, they tended to form really small reproductive population sizes. So they either had no reproductive individuals in the population at the last monitoring date, or they tended to have less than 50 individuals. Interestingly, we tended to find that some faster growing species, particularly some woody subtropical species in the data set, tended to be strong seed producers, and those species tended to form larger reproductive population sizes over time. And so we see this sort of difference in different life forms. So these sort of slow growing, longer-lived herbaceous species that don't invest a lot in reproduction, they tend to not form larger reproductive population sizes. And again, they tend to have longer recruitment lag times to when you get to see seedlings. So these are the species that are at risk of drawing conclusions prematurely on their success. If you only have short-term monitoring data available, you may think these are not performing well. But in reality, what these species are likely doing is investing in below-ground growth, below-ground survival, and that's sort of where they're putting their resources, and they're not putting their resources into reproduction in the early phases of these reintroduction projects. So we also saw the number of threats over time tended to be the most important predictor variable of reproductive population sizes. And so those reintroductions that had more than 2 threats at the time of outplanting, and threats could be anything from invasive species, predators or herbivores at the sites. It could be disease. It could be a number of different factors that were potentially threatening to that species. For those focal species that had 2 or more threats at a given reintroduction site, they were more likely to form reproductive population sizes that were either had no individuals, or they were less likely to form medium to large reproductive population sizes. So they just tended to have a much smaller number of reproductive individuals in their population, which is not surprising because they were experiencing more threats. So threats did turn out to be the biggest predictor over the long term of this particular variable. So I think these analyses collectively have some important implications for best practices in rare plant reintroduction. We're consistently seeing an effect of founder size, and again, some other studies have also found a relationship between founder size and various metrics of success. So I think in terms of best practices, you need to be planting at least 100 individuals out at a site over time to ensure the persistence of that species or that population, and to really overcome small population barriers, those sort of demographic and genetic barriers that may exist in small populations. We also found, again, habitat quality was coming out as an important predictor for population persistence, so it's really important to match the species niche to the reintroduction sites. This is really challenging for many of the rare species we're working with. Many of the G1s and G2s are known from only a handful of sites, and those sites may not be the best indicators of optimal habitat for the species if those habitats have changed over time. And in these cases, if we're unsure of what the optimum habitat is for the species, then we need to use experimentation to sort of understand the species niche and what kinds of sites might be most suitable to support long-term population growth of those species. Threats are important. We need to be reducing threats. We need to understand threats of these rare species, and they need to be taken into account at the time of outplanting. And we're going to need to monitor sites over time for changes in habitat quality. Remember that change in habitat quality turned out to be a really important predictor for the long-term persistence of reintroduced populations. And so we need to be managing and responding to these sort of negative changes over time. And finally, this is really something that's, I think, critical for all reintroductions. You need to have a plan of how you're going to implement the reintroduction, and then you need to have a monitoring plan in place as well, so you can monitor these changes in threats and site quality over time, and then you can adjust your practices accordingly. So I think there's some broader implications from this work as well, and in the data set we showed you today, we had about 275 projects, and about 90% of those projects are persisting on the landscape, at least as of the last monitoring date. Now, I want to not overemphasize that point too much because I think there's some inherent bias within this data set in terms of it probably being biased towards reintroduction projects that perform better than those that perform worse. But nevertheless, for G1 and G2 species, this means there are a large number of individuals in translocated populations that are really persisting in the landscape, and they're increasing the total number of individuals surviving in the wild and the spatial occupancy of these rare species. So what I'm showing you on the left-hand side is the proportion of surviving populations of these 275 reintroduction projects. And survive with a negative and survive with a plus refer to the population trends. So are they declining or are they increasing? And as you can see that a large proportion of reintroductions in Hawaii are actually declining in terms of their population growth over time. And that might be alarming, but I would argue that our analysis points to many of the factors that we need to pay attention to that we can sort of reverse these declines. And I think the fact that we're finding a large number of, well, let me back up. Let me just say our analysis is not showing that any particular species or ecosystem type is more challenging to reintroduce a species into than another one. So we're not finding things like ecosystem type, or there aren't that many species traits coming out as really being important. So it suggests that most species can be reintroduced, but the things that are sort of driving their success rates are really under the control of the manager, the conservation practitioners, and those involved in the reintroduction project. So many of these populations that are declining, I think, could potentially be reversed over time with proper management of both habitat quality and threats at those reintroduction sites. And with that, I want to thank all the sort of dedicated conservationists working to conserve rare species and for sharing lots of data and information for their reintroduction projects. None of this work would be possible without their contributions. So I really want to thank them for all their work. And with that, I'll take any questions.